This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome. Ever wondered how much life on Earth remains hidden in plain sight? This exploration into the fascinating world of cryptic species will reveal how creatures appearing identical can be genetically distinct. We'll uncover the evolutionary secrets behind these masters of disguise and examine the challenges they pose to conservation. Prepare to have your perception of biodiversity transformed as we delve into a world where DNA reveals a reality far more complex than meets the eye. Get ready to discover the astonishing diversity hidden within a world we thought we knew. Chapter 1. The Deceptive Eye. When looks can't tell the whole story. Consider the iconic fly agaric mushroom, Amanita muscaria, a vibrant splash of red and white instantly recognizable in forests across the northern hemisphere. For centuries, this striking fungus was considered a single species. However, the advent of genetic analysis has shattered this long-held assumption, revealing that the fly agaric is actually a complex of several cryptic species, each with a unique genetic makeup, subtly different ecological preferences, and potentially even variations in toxicity. What we once perceived as a single, widespread organism is now understood to be a mosaic of hidden diversity. A similar tale unfolds in the realm of insects. The ruby-spot damselfly, Heterina americana, a common sight along North American waterways, flashes its namesake ruby-red spots as it dances above the water's surface. However, beneath this veneer of uniformity, scientists suspect the presence of a cryptic complex. Subtle behavioral differences, variations in mating calls, and emerging genetic data hint at the existence of at least one other species, masquerading within the familiar form of H. americana. Even charismatic megafauna, like the African elephant, have not escaped the deceptive eye. For decades, these majestic creatures were classified as a single species, Loxodonta africana. But DNA analysis has rewritten their story, revealing two distinct species: the African bush elephant, L. africana, and the African forest elephant, Loxodonta cyclodus. While superficially similar, these elephants possess distinct genetic profiles, occupy different ecological niches, savanna versus forest, and exhibit subtle morphological differences, such as ear shape and tusk size, that were easily overlooked in the past. The Boquart's terrapin, Moramis rivulata, a freshwater turtle inhabiting the Mediterranean region, provides yet another compelling example. Genetic studies have unveiled a complex of several cryptic species within this seemingly homogeneous group. Despite their nearly identical appearances, these turtles are genetically isolated, representing distinct evolutionary lineages that have diverged over time, their differences hidden from view. The world of butterflies offers perhaps the most visually striking examples of cryptic speciation. The genus Heliconius boasts a dazzling array of wing patterns, a testament to the power of natural selection. However, these very patterns, once used to define species, have become a source of taxonomic confusion. Some Heliconius species are so similar that even expert lepidopterists struggle to differentiate them based on morphology alone. Minute variations in wing venation, subtle shifts in color patterns, and differences in the chemical composition of wing scales can indicate distinct species, requiring painstaking observation and sophisticated analytical techniques to uncover. The neotropical skipper butterfly, Astraptes fulgurator, further underscores this point. Initially considered a single, widespread species, genetic testing has revealed a complex of at least 10 cryptic species, each with unique genetic markers and, upon closer inspection, subtle differences in caterpillar morphology and larval food plant preferences. These cases highlight the limitations of relying solely on morphology for species identification. History is replete with examples where morphological similarity has led to misidentification and misclassification, obscuring the true extent of biodiversity. The yellow-spotted, fire salamanders, of the genus Salamandra, once all classified as a single species, S. salamandra, provide a cautionary tale. Genetic analysis has revealed that these salamanders are not a monophyletic group, meaning they do not share a common ancestor. The Corsican fire salamander's closest relative is, surprisingly, the entirely black alpine salamander, indicating that their striking yellow spots evolved independently through convergent evolution. Similarly, the Amazonian frog Pristimantis auckendeni, once considered a single species, was revealed to be at least 3 distinct species that diverged millions of years ago, their morphological stasis masking their deep evolutionary history. The challenge of identifying cryptic species becomes even more pronounced in the microscopic world. The sea slug genus Pontohedyle exemplifies this difficulty. These minute, interstitial creatures, often less than a millimeter in length, exhibit remarkably little morphological variation. However, through detailed examination of their radioli, tooth structures, and genetic analysis, scientists have discovered a surprising diversity of species within this seemingly uniform group. Each species, though morphologically cryptic, possesses a unique combination of genetic markers, radular characteristics, and often subtle differences in internal anatomy. These examples are just a glimpse into the hidden world of cryptic species. As we delve deeper into the genetic makeup of organisms, we are discovering that cryptic diversity is far more prevalent than we ever imagined. This realization has profound implications for our understanding of biodiversity, conservation efforts, and the very way we define species. Just how widespread is this hidden diversity? And what does it mean for our understanding of the tree of life? These questions will be explored in the next chapter, as we uncover the surprising prevalence of cryptic species and the challenges they pose to our traditional view of the natural world. Chapter 2. A World Within a World. Exploring the biodiversity we can't see. Imagine you're an angler, casting your line into one of the many picturesque lakes nestled in the mountains of southeastern Australia. You're after the small, unassuming minnows that dart through the shallows. To you, they all look the same, small, brownish fish, perhaps 1 or 2 different kinds at most. But what if I told you that those minnows, collectively known as mountain galaxias, actually represent a hyper-cryptic complex of at least 15 distinct species? Each lake, each seemingly identical fish, holds a unique piece of the biodiversity puzzle, invisible to the untrained eye but revealed through the power of genetic analysis. This astonishing discovery underscores just how much we might be missing when we rely solely on what we can see. This phenomenon is not confined to the depths of remote lakes. It plays out across all environments and taxonomic groups, from microscopic fungi to charismatic megafauna. Each adapted to its specific habitat. The plant kingdom, too, harbors its share of cryptic secrets. However, identifying cryptic plant species presents unique challenges. Unlike animals, where DNA barcoding using a specific mitochondrial gene is often effective, plants require different molecular approaches. Their genetic makeup makes the animal method unreliable, and scientists have had to develop alternative techniques, such as nuclear ribosomal DNA sequencing, to uncover cryptic plant diversity. This has led to surprising discoveries, like the mottle grass Brachypodium distachyon, a key species in cereal research, which was found to be not 1, but 3 distinct species. Even the common weed, Russian thistle, has yielded a cryptic species hiding within its invasive populations in California. These discoveries highlight the ongoing challenge of deciphering plant biodiversity and the importance of developing ever more sophisticated molecular tools. The insect world, a buzzing hub of biodiversity, is rife with cryptic species. These hidden species can have profound implications for agriculture. Take the whitefly, a notorious agricultural pest. Genetic analysis has revealed that what we once considered a single species is actually a complex of at least 28 cryptic species, each with potentially different responses to pest control methods. This has major consequences for developing effective management strategies. Similarly, some insect parasitoids, used as biocontrol agents, have been revealed to be cryptic species complexes, each specializing on a different host. This complexity can make biocontrol efforts more challenging, as the effectiveness of a particular agent depends on accurately matching it to the target pest species. The very concept of what constitutes a species becomes blurred in the face of cryptic diversity. The term species complex is often used to describe a group of closely related organisms where the boundaries between species are unclear, often due to hybridization or recent divergence. The grizzled skipper butterfly is a prime example. Classified as a superspecies, it's further divided into 3 subspecies, illustrating the hierarchical nature of biodiversity and the challenges of drawing clear lines between species. The existence of cryptic species complicates scientific research in numerous ways. Accurate species identification is essential for understanding ecological interactions, evolutionary processes, and the distribution of biodiversity. But when species look alike, how do we know what we're studying? This is particularly problematic in fields like pest control and disease management, where misidentification can lead to ineffective strategies. Imagine trying to control malaria without realizing that the mosquito you're targeting is actually 6 cryptic species, only 3 of which transmit the disease. This was precisely the problem faced in Europe in the early 20th century, highlighting the critical importance of accurate species identification for effective public health interventions. Cryptic species also force us to rethink how we estimate global biodiversity. If we're missing entire species simply because they look like others, our current estimates of species richness could be vastly underestimated. Some scientists suggest that the true number of species could be as much as 30% higher than currently recognized. This has significant implications for conservation. If we don't know how many species exist, how can we effectively protect them? The discovery of the Atlantic coast leopard frog, a cryptic species found in one of the most densely populated regions of the United States, underscores this point. This frog, only formally described in 2014, was previously overlooked because it was mistaken for other leopard frog species. Its discovery highlighted the need for ongoing research and the potential for finding new species even in well-studied areas. So, what drives the persistence of these hidden species? Evolutionary and ecological factors play a crucial role. Stabilizing selection, where natural selection favors intermediate traits, can maintain morphological similarity even as species diverge genetically. Asexual reproduction, like apomixis in plants, can also lead to the formation of distinct lineages without significant morphological differentiation. These processes demonstrate the intricate interplay between genes, morphology, and the environment in shaping biodiversity. The prevalence of cryptic species underscores a fundamental truth about the natural world. Everything is connected. Species, even those we can't readily see, play vital roles in their ecosystems. They contribute to nutrient cycling, pollination, seed dispersal, and a myriad of other processes that maintain the balance of life. By overlooking cryptic species, we risk misunderstanding the complex web of interactions that sustains our planet. Their discovery reminds us that biodiversity is not just about the number of species, but also about the intricate relationships between them, visible and invisible. Protecting this hidden world within a world is essential for preserving the health and resilience of our planet. Chapter 3. Decoding the Secrets of Life. How DNA is Rewriting the Book of Life. For decades, scientists relied primarily on morphology, the outward appearance of organisms, to classify and differentiate species. But the advent of DNA technology has revolutionized this field, providing a powerful lens to peer into the genetic makeup of life and uncover a hidden world of biodiversity. One of the most striking examples of this revolution comes from the tropical forests of Costa Rica. For years, the two-barred flasher butterfly, Astraptes fulgurator, was considered a single, widespread species, its distinctive wing pattern a seemingly reliable marker of its identity. However, something didn't quite add up. Researchers like Daniel Janssen noticed puzzling variations in the caterpillars of this single species. Some caterpillars sported different colors and patterns, and they seemed to prefer different food plants. These subtle discrepancies hinted at a deeper, hidden diversity. The mystery remained unsolved until the advent of DNA barcoding. When researchers analyzed the DNA of these butterflies, a startling truth emerged. What was once thought to be one species was actually at least 10 genetically distinct species, a cryptic complex masquerading under a single name. The variations in caterpillar morphology and food preferences suddenly made sense. They were reflections of the underlying genetic diversity. This discovery not only revolutionized our understanding of insect biodiversity in the region but also highlighted the limitations of relying solely on morphology for species identification. DNA barcoding has become a cornerstone of modern taxonomy. It works by sequencing a specific region of an organism's genome, typically a gene called cytochrome C oxidase I, COI, located in the mitochondria. This gene is chosen for several reasons. First, it evolves relatively slowly, providing a stable genetic marker for comparing different species. Second, it's found in nearly all animals, making it a universal tool for species identification. Third, mitochondrial DNA is remarkably resilient, allowing scientists to extract and analyze it even from small or degraded samples, a boon for wildlife forensics. The process generates a unique genetic barcode for each species, which can then be compared to a vast reference library of barcodes. If a barcode doesn't match any existing records, it suggests the possibility of a new, undiscovered species. However, DNA barcoding is not without its challenges. One of the main controversies revolves around the threshold of genetic divergence used to define a species. How different do two barcodes need to be to signify separate species? The answer varies depending on the taxonomic group, creating some ambiguity in species delimitation. Another limitation stems from the fact that mitochondrial DNA is inherited solely from the mother. This means that barcodes only reflect the maternal lineage and may not capture the full picture of a species' evolutionary history, especially in cases of hybridization or recent divergence. Molecular phylogenetics offers a complementary approach to unraveling the complexities of cryptic species. This technique uses DNA sequences to reconstruct the evolutionary relationships between organisms, creating a family tree of species. By comparing the DNA of different organisms, scientists can determine how closely related they are and how they have diverged over time. This can be particularly helpful in differentiating between cryptic species that look alike. For instance, the yellow-spotted fire salamanders of Europe, once thought to be a single species, presented a phylogenetic puzzle. Genetic analysis revealed an unexpected twist. The Corsican fire salamander's closest relative was not another yellow-spotted salamander, but the entirely black alpine salamander. This surprising finding indicated that the yellow spots had evolved independently in these two lineages through convergent evolution, a process where unrelated organisms develop similar traits due to similar environmental pressures. Phylogenetics can also reveal cases of morphological stasis, where species diverge genetically but maintain similar appearances over long periods. The Amazonian frog Pristimantis auckendeni exemplifies this phenomenon. Genetic analysis revealed that this species actually represents at least 3 distinct lineages that diverged millions of years ago. Yet, despite this deep evolutionary history, they remain virtually indistinguishable morphologically. Beyond DNA barcoding and phylogenetics, other genetic tools have emerged to further refine our understanding of cryptic species. Metabarcoding takes DNA barcoding to the next level, analyzing entire biological communities from environmental samples like soil, water, or even feces. This technique allows scientists to identify multiple species simultaneously, providing a snapshot of the biodiversity present in a particular ecosystem. It's especially useful in complex environments like the deep sea or dense rainforests, where traditional survey methods may be impractical or ineffective. For example, metabarcoding has been used to identify different salmonid species in rivers, revealing their distribution patterns and uncovering cryptic diversity within these fish communities. Environmental DNA, EDNA, barcoding offers a non-invasive way to detect species by extracting DNA directly from environmental samples. This technique is particularly valuable for monitoring elusive or rare cryptic species, as it doesn't require capturing or disturbing the organisms themselves. EDNA barcoding has even led to the discovery of new species, including previously unknown microbes, fish, and amphibians, further expanding our knowledge of the hidden biodiversity that surrounds us. The implications of these genetic tools are far-reaching. They have not only expanded our understanding of biodiversity by revealing a hidden world of cryptic species but have also challenged traditional taxonomic classifications. The case of the African elephant is a prime example. DNA analysis led to the reclassification of this iconic animal into 2 distinct species, the African bush elephant and the African forest elephant. This reclassification had significant consequences for conservation, as the two species now have different conservation status assessments, reflecting their distinct population sizes and threats. These tools have also provided new insights into evolutionary processes. They have shed light on adaptive radiation, where a single ancestral species diversifies into multiple species to fill different ecological niches, as seen in the hyper-cryptic complex of Australian minnows. They have revealed instances of hybrid speciation, where new species arise through the interbreeding of existing species, as observed in some heliconius butterflies. And they have helped us understand the phenomenon of morphological stasis, where species maintain similar appearances despite genetic divergence, as exemplified by the Amazonian frog Pristimantis auckendeni. The revolution in species discovery and classification continues. DNA technology is rewriting the book of life, revealing a world far more complex and diverse than we ever imagined. Ongoing efforts, like the project to barcode all fish and bird species, promise to further unveil the hidden diversity of cryptic species across the animal kingdom. These tools are not only changing how we classify life but also transforming our understanding of evolution, ecology, and the intricate web of life that connects all living things. The more we learn about this hidden world, the more we appreciate the astonishing biodiversity that surrounds us and the urgent need to protect it. Chapter 4. The Engines of Illusion: Evolutionary and Ecological Forces Behind Cryptic Species. The discovery of cryptic species has unveiled a hidden dimension of biodiversity, a world where genetic distinctions belie morphological similarities. But what evolutionary and ecological forces have conspired to create these masters of disguise? The answer lies in a complex interplay of factors, including adaptive radiation, parallel evolution, recent speciation, hybridization, and the subtle but powerful influence of ecological pressures. Adaptive radiation, a burst of evolutionary diversification, often leads to the formation of what are known as species flocks. These groups of closely related species, inhabiting the same environment, arise when a single ancestral species rapidly adapts to exploit different ecological niches. Darwin's finches of the Galapagos Islands provide a classic example. These birds, descended from a common ancestor, have diversified into a remarkable array of species, each with a beak shape uniquely adapted to its specific food source, from cracking tough seeds to probing for insects. Similarly, the cichlid fishes of the African Great Lakes represent some of the most spectacular examples of adaptive radiation, with hundreds of species evolving in a relatively short period, each specialized to a particular habitat and feeding strategy. Initially, these species were identified based on minute morphological details, such as fin shape, coloration, and jaw structure. However, DNA-based methods have further refined our understanding of cichlid diversity, revealing even more cryptic species within these already diverse flocks. Reproductive isolation tests, such as mate choice experiments, have also confirmed the species status of these genetically distinct but morphologically similar fishes. Parallel evolution, a separate but equally fascinating process, occurs when unrelated species independently evolve similar traits in response to similar environmental pressures. This can lead to the formation of cryptic species when the shared traits mask underlying genetic differences. Imagine two butterfly species, one in South America and one in Asia, both evolving strikingly similar wing patterns to deter predators. To the casual observer, they might appear to be the same species, but their DNA tells a different story. They are unrelated lineages that have converged on the same solution to a shared ecological challenge. Similarly, unrelated fish species inhabiting fast-flowing streams in different parts of the world may independently evolve streamlined body shapes to cope with the current. These examples demonstrate how parallel evolution can create the illusion of shared ancestry, leading to the underestimation of true biodiversity. Recent speciation, the relatively recent divergence of species from a common ancestor, further complicates the picture. When species have only recently split, there may not have been enough time for substantial morphological differences to evolve, resulting in cryptic species. Ring species, such as the Ensatina salamanders of California, provide a compelling illustration of ongoing speciation. These salamanders form a ring-like distribution around a central valley. Adjacent populations can interbreed, but populations at the opposite ends of the ring are reproductively isolated, effectively representing distinct species. This gradual transition from one species to another along the ring highlights the difficulty of drawing clear species boundaries and the dynamic nature of speciation. The study of intraspecific variability, the variations within a species, can offer valuable insights into this process, providing clues about how populations are diverging and potentially setting the stage for future speciation events. Hybridization, the interbreeding of different species, adds another layer of complexity to cryptic species evolution. Reticulate evolution, a web-like pattern of ancestry resulting from hybridization, can lead to the formation of hybrid species with intermediate characters, blurring the lines between parental species. The heliconius butterflies, renowned for their vibrant wing patterns, provide striking examples of hybrid speciation. In some cases, hybrid heliconius species have evolved novel wing patterns that differ from both parental species, while in other cases, they may exhibit intermediate patterns, making them difficult to classify within the species complex. This phenomenon is not limited to insects. Hybrid speciation has also been observed in fungi and plants, creating taxonomic challenges and highlighting the dynamic and interconnected nature of species boundaries. The classification of hybrid plant species, often referred to as nothospecies, particularly in groups where polyploidy, having multiple sets of chromosomes, and apomixis, asexual reproduction through seeds, are common, further complicates the issue. Ecological factors also play a significant role in shaping cryptic species diversity. Stabilizing selection, a force that favors intermediate traits, can maintain morphological similarity in species complexes adapted to specialized environments. For example, symbionts, organisms that live in close association with a host species, may experience stabilizing selection that preserves their similarity to the host, even as they diverge genetically. This is seen in some gut bacteria, where maintaining a specific shape is essential for their interaction with the host's digestive system. Similarly, species adapted to extreme environments, such as deep-sea hydrothermal vents, may experience stabilizing selection that preserves traits essential for survival in these harsh conditions, even as they diverge genetically from related species in less extreme environments. Asexual reproduction can also contribute to cryptic species formation. Apomixis in plants, where seeds are produced without fertilization, allows lineages to diverge genetically without the mixing of genes that occurs in sexual reproduction. This can lead to the accumulation of genetic differences and the formation of cryptic species that are morphologically indistinguishable. Other forms of asexual reproduction, such as budding or fragmentation, can also lead to similar outcomes in other taxonomic groups. Morphological stasis, the long-term preservation of similar morphology in distinct species, provides a fascinating counterpoint to the dynamic processes of adaptive radiation and recent speciation. Stabilizing selection, as seen in host-specific symbionts and species adapted to extreme environments, likely contributes to this morphological stasis. The yellow-spotted, fire salamanders, of the genus Salamandra, where convergent evolution has produced similar coloration patterns in unrelated species, offer another example of how morphological similarity can obscure underlying genetic diversity. These cases highlight the importance of genetic analysis in uncovering the true extent of biodiversity and understanding the complex evolutionary processes that shape it. The story of cryptic species is a testament to the power of evolution and the interconnectedness of life. It challenges us to look beyond the superficial and delve into the genetic code to unravel the true diversity of the natural world. As we continue to develop and refine genetic tools, such as DNA barcoding and metabarcoding, we are poised to uncover even more hidden species, further enriching our understanding of the intricate web of life on Earth. The next chapter will explore these powerful tools in more detail, revealing how they are being used to decode the secrets of life and rewrite the book of biodiversity. Chapter 5. Guardians of the Unknown, protecting the species we're just beginning to understand. The natural world, as we've seen, is a master of disguise. But the illusions cast by cryptic species, organisms that appear nearly identical yet are genetically distinct, pose a unique challenge for conservation. How do we protect what we can't readily see, what we're only just beginning to understand? Traditional conservation, often reliant on easily observable characteristics, struggles to address the nuanced needs of these hidden forms of life. One of the most pressing issues is the scarcity of data. Cryptic species, by their nature, are elusive subjects of study. Their hidden diversity often translates to a dearth of information about their distribution, population size, ecological roles, and the specific threats they face. This lack of knowledge makes it difficult to assess their conservation status and develop effective strategies for their protection. A species once considered common and widespread might, upon closer genetic inspection, reveal itself to be several distinct species, some with much smaller populations and restricted ranges, placing them at considerably higher risk of extinction. Consider the Jonas mouse lemur, a small, nocturnal primate recently discovered in the forests of Madagascar. While its existence was only recently confirmed, this tiny lemur is already teetering on the brink of extinction. The fragmentation of its forest habitat due to logging and slash-and-burn agriculture, coupled with the pressures of the illegal pet trade, has pushed this species to the edge. Similarly, the Popa langur, a newly described primate from Myanmar, faces a precarious future. Confined to a small area of forest fragmented by agriculture and human settlements, this langur's population is dwindling, threatened by habitat loss, hunting, and the ever-present risk of genetic isolation. The clouded tiger cat, a newly recognized species within the Oncilla complex, underscores the importance of considering cryptic diversity and conservation. This small, spotted cat inhabits the rapidly disappearing cloud forests of Central and South America. Protecting this species requires not only preserving remaining cloud forest fragments but also implementing strategies to connect these isolated patches, allowing for gene flow and increasing the resilience of the clouded tiger cat population. Efforts to combat deforestation, promote sustainable agriculture, and reduce human encroachment are crucial for the long-term survival of this elusive feline and the unique ecosystem it calls home. Even in seemingly well-studied regions, cryptic species can remain hidden in plain sight. The Atlantic coast leopard frog, found along the densely populated eastern coast of North America, was long mistaken for other, more common leopard frog species. Its true identity was only revealed through a combination of genetic and bioacoustic analyses. This delayed recognition has had significant implications for conservation efforts. Previous assessments of leopard frog populations in the region likely overestimated their numbers and underestimated the vulnerability of this distinct species. Now, armed with a better understanding of its true identity and distribution, conservationists can develop targeted strategies to protect this previously overlooked amphibian. The challenges of applying traditional conservation methods to cryptic species are numerous. Morphological similarity can lead to misidentification, hindering accurate population assessments and potentially misdirecting conservation resources. For example, imagine managing a fishery based on the assumption of a single, abundant fish species, only to discover later that it's actually a complex of several cryptic species, some of which are rare and declining. This scenario underscores the importance of incorporating genetic data into conservation assessments. The discovery of cryptic species also has profound implications for resource allocation. Limited conservation funding must be strategically distributed to protect the greatest number of species and maintain ecosystem integrity. When a species is revealed to be multiple cryptic species, each with its own specific needs and threats, conservationists must re-evaluate priorities and adjust management strategies accordingly. This can be particularly challenging when some of the newly recognized species are highly endangered and require immediate intervention. Integrative approaches, combining genetic data with ecological and morphological information, are becoming increasingly crucial for cryptic species conservation. For instance, researchers studying the sea slug genus Pontohedile have combined genetic analysis with detailed examination of their radular morphology and internal anatomy to distinguish between cryptic species. This integrated approach has allowed them to identify and describe new species within this morphologically conservative group, providing a more accurate picture of its true diversity. New identification and survey techniques are also being developed. Environmental DNA, EDNA, analysis, for example, allows scientists to detect the presence of a species by extracting DNA from water, soil, or other environmental samples. This non-invasive method is particularly valuable for surveying cryptic species that are difficult to capture or observe directly. Acoustic monitoring, using specialized recording equipment to capture and analyze the calls of different species, is another powerful tool for identifying cryptic species, particularly in groups like frogs and insects, where vocalizations play a key role in mate recognition and species differentiation. Habitat and landscape-scale conservation initiatives are essential for protecting cryptic species. Protected area networks, designed to encompass a representative range of habitats and ecosystems, can safeguard cryptic species even when we lack detailed information about their specific distributions and requirements. For example, conservation efforts in the eastern Arc Mountains of Tanzania, a biodiversity hotspot recognized for its high levels of endemism, including many cryptic species, focus on establishing and managing a network of protected areas to preserve the region's unique flora and fauna. Local communities and indigenous knowledge can play a vital role in cryptic species conservation. Indigenous peoples often possess a deep understanding of the natural world, including the subtle differences between seemingly identical organisms. Their knowledge can be invaluable for identifying cryptic species, understanding their ecological roles, and developing culturally appropriate conservation strategies. For example, indigenous communities in the Amazon rainforest have long recognized distinct varieties of medicinal plants that were later confirmed to be cryptic species through scientific analysis. Citizen science initiatives, where volunteers contribute to data collection and monitoring, offer another avenue for expanding our knowledge of cryptic species. Projects like the Consortium for the Barcode of Life, which aims to create a DNA barcode library for all species, rely heavily on citizen scientists to collect and submit samples. These initiatives not only generate valuable data but also raise public awareness about the importance of biodiversity and the hidden world of cryptic species. Protecting the species we're just beginning to understand requires a fundamental shift in our approach to conservation. We must move beyond relying solely on what we can see and embrace the power of genetic tools and integrative approaches to unravel the hidden tapestry of life. By fostering collaboration between scientists, local communities, and citizen scientists, we can build a more comprehensive understanding of cryptic species and develop effective strategies to ensure their long-term survival. The future of conservation depends on our ability to become guardians of the unknown, protecting not only the charismatic creatures that capture our imaginations but also the hidden species that play essential roles in the intricate web of life on Earth. Cryptic species, genetically distinct yet morphologically similar, challenge our understanding of biodiversity. DNA barcoding and phylogenetics reveal hidden diversity across taxa, from butterflies to frogs, impacting conservation, taxonomy, and evolutionary biology. This hidden diversity underscores the limitations of morphology-based identification and reveals underestimated species richness. These findings have profound implications for ecosystem management and conservation strategies. Where will this genetic revolution lead us? Share your thoughts and questions below. Like, subscribe, and suggest future topics. Never stop exploring the wonders of the natural world. There's always more to discover.